Introduction. Biofouling refers to the undesirable accumulation and growth of microorganisms, plants, algae, and animals on surface immersed in water. An antibiofouling agent refers to substance or coating designed to prevent the accumulation of unwanted biological materials such as bacteria, algae, and other microorganisms on surface exposed to natural environments like water. Biofouling is a biological phenomenon undesirable for the shipping industry, both from an economic and environmental point of view. The accumulation of biofouling organisms on the hull surface causes deterioration of the ship's hydrodynamic performance by imposing a gradually growing drag penalty. The objective of this project is the synthesis of zinc, curcumin, and silver-coated polymeric material to mitigate biofouling in marine structures, and to conduct series of tests such as colony count and distribution to study the efficiency and durability of the coating. These are the workflow in which the project work is going to be. And initially the selection of polymer, antifouling agent, and solvent. Then using magnetic stirrer we stir the combination, preparing the antifouling coating, heating in the vacuum oven for the settling of coating on the stainless steel specimen, and characteristic study, optimization recommendation, documentation record, then at last the result. The chemical components required are polyvinyl alcohol is a polymer, polyethylene glycol polymer, and dimethyl sulfoxide is used as a solvent, and the salts added are zinc chloride, curcumin, and silver nitrate. Polyvinyl alcohol is a synthetic polymer that belongs to the polyvinyl ester family, is known for its biocompatibility, meaning it's less likely to harm marine life compared to certain biocides, and is generally considered to be less toxic compared to some traditional antifouling agents. Polyethylene glycol has been shown to resist protein adsorption, thereby reducing the initial step in biofilm formation. PEG is biocompatible, meaning it is non-toxic and does not elicit an immune response when in contact with biological tissues. Zinc has inherent biocidal, antibacterial, and anti-algal property which helps prevent the attachment and growth of marine organisms. Zinc-based antifouling coatings are effective in range of marine environments including salt water and brackish water. Curcumin is nothing but the purest form of turmeric, and curcumin could potentially be used to create synergetic effects, enhancing the overall performance of the coating agent, a broader range of fouling organisms. And curcumin is a natural compound and is expected to be more biodegradable compared to silver ions from Ag+ released from silver coating exerts antimicrobial effects by disrupting microbial cell membranes, interfering with cellular functions, and inhibiting microbial growth. Silver coating can provide long-term antimicrobial protections even in challenging environments. In scanning electron microscopy testing, the SEM images of the top surface of the specimens, coated specimens were taken, in which PVA, PEG, where only polymer coating is done, shown a clean image compared to the other silver, curcumin and zinc, in which the particles of the respective salts were shown. In atomic force microscopy testing, the surface roughness of the coated samples were analyzed, and silver coated sample has high roughness compared to the other three samples, which the lower the surface roughness, the hydrophobic nature of the from FTR testing results, we can understand the chemical bonds formed between the chemical compounds while preparing the antibiofouling coating, and in which the frequency in which the chemical bonds were formed. From this we can analyze how stable the chemical bonds are formed, and in this case there are formation of OH alcohol bond, and alkanes and sulfoxide bonds were formed. In antimicrobial testing, the biofilm forming bacteria and fungals were taken for the zone inhibition test. As we see in the first antifungal testing, there were 4 samples were taken. One is polyvinyl alcohol alone, and another 3 are polyvinyl alcohol with silver, curcumin and zinc. And in antifungal, for fungal activity, we took Candida albicans. In that test, it shows that curcumin and AG shows higher antifungal characteristics than zinc and normal polyvinyl alcohol. And for bacteria, we took E. coli and SRS. And for E. coli, silver, curcumin and zinc based coatings are shown highly efficient against E. coli. And in SRS2, the silver and curcumin and zinc formed better antibacterial characteristics. As the zone formed, the larger the diameter of the zone, the higher the effective against the microbes. Advantages: use of natural antibiofouling agent will act as toxic free and eco-friendly. Increase the ship's hydrodynamics by preventing the biofouling formation on the hull of the ship, not only in ships but also in marine structures that are immersed in water, and also the sensors used in ships that are immersed in water, which reduces efficient by the formation of biofilm. Applications: antifouling coatings are extensively used on ship hulls to prevent the growth of marine organisms. Antifouling coatings are applied to underwater structures such as offshore oil platforms and underwater pipelines to mitigate biofouling. Antifouling coatings are used on desalination plants to prevent the buildup of biofouling. Thank you.